The debate in front of us is, let it be resolved, that animal models are an outdated paradigm for discovering cures and treatments for human diseases and testing drugs. And Dr. Greek will be the first speaker and will present his case. Dr. Greek.
How was that? Can you hear me? Maybe push it up a little bit further. Testing 1, 2, 3, 4. Is that good? Is that good in the back? Is the sound guy still here? Is the sound guy still here? I don't think it's on. It's on. There we go. Is that better? I don't know how to adjust it.
Probably in your collar.
Testing 1, 2, 3, 4. Is that better? I'll speak loudly. All right. You can use the other microphone.
Be careful with feedback, you can do this.
I'll tell you what, let's turn this one off.
Yeah. Right. Whiz it over.
Because your mouth is closer to
Okay, how's that? That good? All right. Okay, well, before I begin, I would like to thank the University of Buffalo for having me in the honors program, of course. I'd also like to mention the UB Spectrum for writing that very nice article. Steabon, the reporter, did a very nice job on that. I appreciate it. Of course, I'd also like to thank the Center for Inquiry and Andrew Skolnick for participating. David Trickle for being brave enough to be the moderator. Of course, I would not be here were it not for Dr. Nobis, and truly, they were very kind and allowed me to spend the night with them last night, and I do appreciate that. And finally, I'd like to thank you, the audience, for giving up your Sunday afternoon to come listen to a blowhard talk about scientific stuff. So, thank you. Now, the first slide, it says in summary what I'm going to try to explain in a very limited amount of time. And this is kind of science-y stuff, and if you don't have a science background, I do apologize, but this is really the heart of the argument. And the slide says, because of differences in gene regulation and gene networks vis-a-vis evolutionary and molecular biology, we now understand why even two nearly identical complex systems, say a chimpanzee and a human, or even identical twins, may respond differently to the same stimuli, and hence why one complex system, or species, cannot reliably predict response for a different complex system, or species. Current biomedical research is studying disease and drug response at the level where the differences between complex systems, be they two different species or even two different humans, manifest. Hence, using animals as causal analogical models for human disease and drug testing is a scientifically invalid paradigm. And at the end of the lecture, we're going to see that same slide again, and hopefully you'll know what all those little words mean by then. Our second slide, before we talk about using animals as causal analogical models, I need to define our subject a little bit better. Animals can be used in roughly 9 different ways in the pursuit of scientific knowledge. Now, what I am going to be addressing today is only numbers 1 and 2, and you might want to refer back to this slide on your handout as I continue the lecture. I am going to be addressing the use of animals as models for human disease and animals as test subjects or models for drug testing. And I'm going to claim that is scientifically invalid. Now, that leaves us 7 ways that animal rights people might object to to animals being used, but these 7 ways are scientifically valid. For example, you can use animals as spare parts. I'm from California. Our beloved governor recently had an aortic valve replacement, and where did that valve come from? A pig. Yes, I've heard all the jokes. Animals can be used as factories. For example, animals were used for decades as a source of production for insulin, and many lives were saved because of cow and pig insulin. Animal tissue can be used to study basic physiological principles. Animals can be used for dissection. They can be used as a heuristic, and so on and so on. All of these, numbers 3 through 9, are scientifically valid, okay? But they are not what we call causal analogical models. That's what CAMs means. And it will be my contention that when animals are used as CAMs, that is scientifically inappropriate. Okay, there's 5 parts to the lecture, and again, I apologize because I've got to go pretty fast. There's 5 parts to the lecture, and the first part is that physics sets the standard for science. Sir Isaac Newton said many years ago, he said, quote, "Therefore, to the same natural effects, we must, as far as possible, assign the same causes." And he went on to say that this rule applies to respiration in man and beast and the descent of stones in Europe and America. Now, Newton and his follower, Claude Bernard, who really made animal experimentation what it is today, he's really the father of modern-day animal experimentation, believed in what is called the resolutio compositive method of study, or what we might today call the method of analysis and synthesis. And this revolves around causal determinism. Again, if something caused X to happen in Europe, that same thing would cause X to happen in the U.S. Well, Newtonian physics is and was a wonderful paradigm. But other paradigms have come along since Newton, including relativity, quantum theory, chaos theory, complexity theory, which we're going to discuss a little bit, and string theory. Now, none of these things have replaced Newtonian physics, but they have expanded on the way we look at the physical world. So, let's talk a little bit about what complexity is. Today, you see a lot in the press, in the literature, both scientific and lay literature, about complex systems or complexity. And these are some of the characteristics of a complex system. Emergence is one. Of interest to us, perhaps, is the fact that relationships are not linear in complex systems. A very small stimulus, for example, may cause a very large effect, or it may cause no effect at all. And extrapolating from one complex system to even an almost identical complex system is very difficult. The relationships contain feedback loops, and most importantly, vis-a-vis reductionism and determinism, the parts cannot contain the whole. Newtonian physics was all about reductionism and determinism, and that was what Claude Bernard took from Newton and applied to research with animals. And it worked very well for a while, but as we will see as you study complex systems, like this one, the paradigm fails. Now, the causes, this is a complex system. This is actually a gene in a fruit fly. And you can see there's a lot of lines that mean that these genes interact with each other. There's feedback loops. Sometimes you can delete one of the genes and have no effect at all. Sometimes you can delete the gene and you see a huge effect. This is a diagram of a complex network. The causes and effects of the events that a complex system experiences are not proportional to each other. The different parts of a complex system are linked to and affect one another in a synergistic manner. There is positive and negative feedback in a complex system. The level of complexity depends on the character of the system, its environment, and the nature of the interactions between the system and the environment. And again, this leads us back to nonlinearity. Now, in the past, society has learned to understand reality through simplification and analysis vis-a-vis reductionism, and it worked great. Some important simple systems are successful idealizations of primitive models of particular real situations. For example, a perfect sphere rolling down an absolutely smooth slope in a vacuum. Now, this is the world of Newtonian mechanics, and it ignores a huge number of other simultaneously acting factors. Although it might sometimes not matter that details, such as the motions of billions of atoms dancing inside the sphere's material, are ignored, in other cases, reductionism may lead to incorrect conclusions. Now, this is especially true in complex systems. In complex systems, we accept that processes that occur simultaneously on different scales or levels are important, and the intricate behavior of the whole system depends on its units in a non-trivial way. Here, the description of the entire system's behavior requires a qualitatively new theory, because the laws that describe its behavior are qualitatively different from those that govern its individual units. What we are witnessing in this context is a change of paradigm in our attempt to understand the world. The laws of the whole cannot be deduced by digging deeper into the details. And there's a name for this. It's called systems biology. And Leroy Hood and others are founders of this field. So, as with chaos theory, the behavior of a self-organizing complex system cannot be predicted by studying a different system. And complex systems do not observe the principles of additivity. And what I mean by that is their components cannot be divided up and studied in isolation vis-a-vis reductionism. As I said, the causes and effects of the events that a complex system experience are not proportional to each other. The different parts of a complex system are linked and affect each other in a synergistic manner. And I hope you can kind of understand what I said when you look at that system of genes and how they interact. If you can't, I brought another slide that looks remarkably like the first one. Okay. Now, living systems, such as chimpanzees, mice, and humans, are obviously examples of complex systems. It should be equally obvious, therefore, why extrapolation between species will be problematic. Small changes on the genetic level can lead to very large differences between species. Indeed, that's what evolution is. So the claim that humans and rats are the same animal, just dressed up differently, simply isn't true. Moreover, it is irrelevant to point to observed similarities in genetic makeup between species, since the details of the differences are in the interactions between the conserved genes, not in the genes themselves. It is as though humans and rats have a common genetic keyboard on which different phenotypic tunes are being played. What matters is not the similarity with respect to the keyboards, but the difference with respect to the order and timing of the activation of the keys. Predicting human response based on an animal model is not an example of applying a relatively simple set of well-established scientific principles as we see with Newtonian physics. Indeed, living organisms are much better examples of complexity theory than Newtonian physics. Using the example of a model airplane, as so many defenders of the animal model do, is a good example of this. Studying a model airplane, especially a paper airplane, will allow the observer to demonstrate the basics of flight. Bernoulli's equations hold true. But if anyone seriously believes this method can be or is used to build or repair a 747, they're deluded. Just as animal models can be and indeed were used to demonstrate very basic facts concerning anatomy and physiology, so a model plane can be used to demonstrate basic physics laws concerning flight. But today, when we want to know why a 747 crashed, we don't build paper airplanes, and neither should we suggest to the public, who is paying for animal experiments, that a cure for AIDS, cancer, or stroke will be derived from animal models. Now this leads us to part 3, which is the regulation and expression of genes. The mouse, we know, has about 30,000 genes. We also know that humans have about 30,000 genes, and what's more, 99% of those genes are shared. And what I mean by that is humans have about 300 genes that mice don't have, and mice have about 300 genes that humans don't have. Mice and humans both have the same gene that when activated in mice grows a tail. Okay? in humans, perhaps with the exception of myself, don't grow a tail. I've been accused of having horns and a tail. But most humans do not grow a tail even though they have this gene. And the reason they don't is because the gene is not turned on. Now, in the old days, if you look to the left of the diagram, this is how we thought biology worked. A single input, whether it was the environment or a genetic switch or a genetic signal, led to a single output. Today, because of the Human Genome Project and other research, we know that genetic networks actually control what goes on in the body. And these genetic networks have multiple inputs and multiple outputs. And even if you have a genetic network that is identical in a mouse and in a human, because of all this interaction that's involved, you can get two entirely different outcomes from the same stimulus. Not only that, but because of very small differences in just one gene, say the gene at the top, you can have a cascade effect that will turn the stimulus into something very different when it gets to the bottom. So genes work in networks, they're interconnected, one influences another, and any small change at the appropriate time will result in a cascade effect that will have major implications. Now this is kind of a little mathematical formula of what I've just said. If you have inputs or stimuli or genes, A sub 1 through A sub n, that are interacted on by environmental factors or other genes for that matter, as represented by B, you can have multiple different outcomes, even though you start with the same conditions. Well, here's what that means. This is a slide that was in Drug Discovery Today. It's a magazine that relates to drug development. And this was a slide that was done by Dr. Bob Coleman in his article. And what he looked at was one gene, it's the CFTR gene, and he assayed the amount of this gene that was turned on in mice and in humans in all these different tissues. Now, as you can see, in every single tissue, the gene is present. But in the mouse, except for the salivary glands, which is the first one, in the mouse, it's turned on far greater, sometimes by 10,000-fold, than it is in the human. Okay? This is the difference between you and a mouse. Okay? Same gene, regulated and expressed differently. Now, the Public Library of Science has a new journal called Biology, and in an editorial, they said this about mouse models' autoimmune diseases. And they were basically talking about all the things that I've talked about so far. They said, these results fall in line with mounting evidence that background genes are not silent partners in gene-targeted disease models, but can themselves facilitate expression of the disease. This finding underscores the notion that genes are not solitary, static entities. Their expression often depends on context, with genetically complex diseases having the requisite combination of susceptible genes does not always lead to the disease. And again, we get back to the network. Same network in you, same network in me. I may suffer from cancer, you may not.